silent mutation, you still get a serine. For a nonsense mutation, you get a stop codon. For a missense mutation, we got a leucine instead of a serine. And they're in different biochemical groups. This is a serine is a polar amino acid, and leucine is a nonpolar amino acid. And so this is why you guys need to know the biochemical groups. The last type is a neutral mutation. It has a different amino acid, but the amino acid is in the same biochemical group, so it's not the same as missense. So missense is a different amino acid that's in a different biochemical group, and when you're writing your answers on your ice, you need to say different amino acid and different biochemical group, or different amino acid but in the same biochemical group. And your test answers need to say that too, or they will be marked wrong. So DNA-based substitutions affect translation of the polypeptide. So here you've got a transcript that is being translated. You get a nice full-length functional protein. If instead you have a missense mutation caused by a base substitution, you're going to get a shorter protein, a truncated protein. Termination of translation, this is a non-functional protein. So this is how base substitutions can affect translation of your polypeptide. Does each anticodon recognize one and only one codon? What is an anticodon? Well, you should know. It's in the tRNA, and it is anti, or opposite of the codon. So it's a tRNA, RNA, messenger RNA interaction between the codon and the anticodon. The third position can be different. The third position of the codon, which is the first position of the anticodon, can be different. So G doesn't usually go across from U. But we have something called the wobble hypothesis. Not all point mutations change the amino acid structure. So even though this is a base substitution, we have, instead of a C, we've got a U, we're still getting the right amino acid, serine. So this is an example of a silent mutation. And so wobble hypothesis can allow for silent mutations. This is the codon chart. You will be provided this on your exam. You should use this for your ICE. So you can see, for example, two codons, UUU and UUC, both call for phenylalanine. We've got our stop codons here, right? You guys have done this in previous classes before. It's the start codon here. So here's A, U is the second nucleotide, and then the third is within the cell. You can see, for example, that serine has these two codons right here, AGU, AGC, and it also has these four codons that call for it, UCU, UCC, UCA, UCG. So the code is degenerate. There's you can have these differences, especially in the third nucleotide as shown by this cell here with all of these four codons that call for the same amino acid. You can have a mutation, but it's still going to call for the same amino acid, and if it's the same amino acid, it is a silent mutation. From this table 15.2, I want you to remember this. It's showing that the first position of the anticodon, I, inosine, can pair with the third position of the codon, A, U, or C. This is shown right here. So if the codon is 5' to 3', then anti-parallel rule, we have to have the 5' of the anticodon opposite of the 3' of the codon, and the I is shown here, this inosine, which is only found in the tRNA, not the messenger RNA, can base pair with either A, U, or C. So this is part of how we get this wobble in the code. This is the one thing I'm asking you to remember from this table which talks about the wobble hypothesis. Concept check. Through wobble, a single blank can pair with more than one blank. The answer is a single anticodon can pair with more than one codon. So if you go back to that chart, we had all these different codons. They brought us the same amino acid. So that meant that the same anticodon could pair with more than one codon. So here's one codon, UCC, here's the other one, UCU. The same anticodon, AGG, is pairing with both of those. So let's go back to this. The same anticodon can pair with more than one codon. And the answer is D. So the summary is Bowen-Conradi syndrome and inherited congenital asplenia are ribosomopathies. Classical experiment we looked at with Beadle and Tatum. One gene, one enzyme in biochemical pathway. Concept we focused on was mutations at the DNA level may change the amino acid code that is translated, potentially affecting the function of the polypeptide, frame shift, missense, nonsense, or not. We could get a silent mutation as well. So that's the end of this portion of the translation lecture for chapter 15. The next lecture will focus on the 3 sites that are found in the ribosome: the A site, the P site, and the E site.